Hello dear students, welcome to this online video lecture. Myself Pawar M.A. from Asian College, Karad. I am conducting here this video lectures under the Rayat Online School Education, that is the ROSE project. I am conducting here first lecture from biology. The topic is plant growth and mineral nutrition. So let's start. Content at a glance. First point is plant growth. Second, phases of growth. Condition for growth. Growth rate and types of growth. Next point is growth curve. Next, differentiation, dedifferentiation and redifferentiation. Next point is development, plasticity, growth hormones, photoperiodism, vernalization, mineral nutritions, and last point is nitrogen cycle. These are the 13 points are present in this topic, plant growth and mineral nutrition. So let's start. First point is plant growth. What is mean by growth? Growth can be defined as the increasing of plant volume or mass with or without formation of new structure such as organs, tissues, cells or cell organelles. Growth is usually associated with development and reproduction. So, growth is one of the characteristics features of living organism. Next, growth as a phenomenon has a two aspects, quantitative and qualitative. Quantitative aspect speaks for an increase in length, breadth, size, volume, body, mass or dry weight and a number of cells. Growth as a quantitative changes in a final and product of successive metabolism. Moves to the next slide. Qualitative aspects talks about the changes in the nature of growth where development is an ordered change or progress while differentiation leads to higher and more complex state. Growth thus can be defined as permanent, irreversible increase in the bulk of organism accompanied by change of form. In multicellular plants, means vascular plants, growth is indeterminate and occurs throughout the life indefinitely. Moves to the next slide. It is restricted to some specific region called a meristem, which are the regions where new cells are constantly and continuously produced. Meristems are of three types of on location: apical meristem, InterCaliber Meristem and Lateral Meristem. These are the types of meristem. The picture shows apical meristem, InterCaliber Meristem and a Lateral Meristem on the basis of these locations and these three types of meristem. Moves to the next slide. Apical meristem. In vascular plants, growth is restricted to the apices of root and shoot. It is responsible for growth in length, height and the differentiation or cell type. It contributes to the primary growth. Apical meristem is primary growth and these are the apical meristem. The picture shows root apical meristem. These are the root cap and the apical meristem. Moves to the next slide. Second one is InterCaliber Meristem. It is located at the node or at the base of internode of stem. Next, it is primarily responsible for increasing length of internodes and also for formation of leaf primordia and lateral bud. These are the InterCaliber Meristem. The picture shows InterCaliber Meristem. This zone of differentiation and this zone of elongations. These are the epidermis, root cap, apical meristem, endodermis, cortex, vascular cylinder, xylem, phloem, and root hair. The picture shows InterCaliber Meristem. Moves to the next slide. Lateral Meristem. These are the third meristem type. It is located laterally along the axis of dicotyledons and gymnosperm. It is located as a strip in the vascular bundles of stem of dicots. It is called as a vascular cambium. It is responsible for increase in the girth of the stem due to addition of secondary vascular tissue. The picture shows lateral meristem. These are the cortex region, vascular bundles means xylem and phloem, fascicular cambium, interfascicular cambium, also known as vascular cambium, cork cambium, and cork. These are pith region, and the lateral meristem responsible for growth in width or girth, secondary growth, and vascular cambiums. These are the lateral meristem. Moves to the next slide. Do you know root and stem shows indefinite or indeterminate growth. However, in organs, leaves, flowers, fruit, growth is determinate. That is, they grow up to a certain genetically distinct size. In unicellular plants, growth is uniform and determinate. So, next point is phases of growth. The cells in the meristem divide, enlarge, and get differentiated. Corresponding to these three stages, there are three phases of growth. These are the three phases of growth. Phase of cell division or formation, phases of cell enlargement and elongation, next phases of cell maturation and differentiation. These are the three phases of the growth. Moves to the next slide. First phase is a phase of cell division or formation. Cells of meristems are thin-walled, non-vacuolated, having prominent nucleus and granular cytoplasm. Meristem cells undergoes mitotic division to form two new cells. One cell remains meristematic and the other cell undergoes enlargement of differentiations. In this phase, rate of growth occurs at a slower pace. It is lag phase. And mitotic, you know the mitotic and meiotic divisions. It is a reductional or equational divisions. These form in the phases of the cell divisions. Moves to the next slide. Next phase is phase of cell enlargement or elongations. These are cells are elongated or enlargement. The newly formed cell becomes vacuolated, osmotically active, and turgid due to absorptions of water. The turgidity results in the enlargement of cell both lengthwise and breadthwise. This turgidity of results in enlargement of cell both is lengthwise or breadthwise. Next, in these phases, new wall materials and other materials are synthesized to cope up with the enlargement, and the growth rate in this phase occurs at the accelerated pace, exponential or log phase. The first phase is a lag phase or second phase of cell elongation or enlargement. It is the log phase or exponential phase. Moves to the next slide. Now, third phase is phases of cell maturation or differentiation. The enlargement cell now becomes specialized to perform specific function and attains maturity, both morphological and physiological. In this phases, rate of growth slow down and comes to a steady state or stationary phase. This phase is also called as a stationary phase. First phase is cell lag phase, second phase is log phase, and third phase is stationary phase, means the phases of cell maturation or differentiations. Moves to the next slide. The picture shows phases of growth in a root position of a radical at the beginning and attain. These are the plumule, this zone of mature cell, zone of cell differentiations, zone of cell elongations, and zone of cell formations. These are the phases of growth in root. Moves to the next slide. The third point is condition for growth. The different environmental and physiological conditions necessary for the growth includes water, supply of nutrients, temperature, oxygen, carbon or nitrogen ratio, gravitational force, light and growth hormones. The chief conditions are explained below. Water is the essential component of protoplasm and maintains turgidity of the cell. Do you know the water is very important or essential component for the growth in any plants? It acts as aqueous medium for biochemical reactions. Moves to the next slide. The conditions for growth is a physiological conditions, environmental conditions are included in plant growth. Moves to the next slide. Microelement and macroelements are nutrients required for the proper growth of the plant. These two elements are required for proper growth of plants. Optimum temperature ranges between 25 to 35 degrees Celsius. Oxygen is essential for respiration and release of energy. Light is very much essential for germination of seed and photosynthesis. It is also important part of the germinations of the seeds. Light is very important, oxygen is very important component. These are the components are very important for the growth. Gravitational force decides their directions of growth of the shoot and the root. These are very important, gravitational force, because they decided directions of growth, shoot or root. Moves to the next slide. Next point is growth rate and types of growth. But growth is a various method of measurement of linear growth of structure and radical. The direct method. These are the methods of growth. Direct method, horizontal microscope, oxynometer, chrysograph. These are the measurement of linear growth and stem and radical. The first is direct method. It can be easily be measured with the help of ordinary measuring scale. It is a simple method. Direct method is it is a simple method because they are measured with the help of ordinary measuring scale. Next is horizontal microscope. It is used as a measure of growth in fields. Next, oxynometer. These equipments is used for precise measurement of linear growth of shoot. Next, chrysograph. It is records primary growth very accurately. It magnifies growth up to 10,000 times, giving information of growth per second. It is developed by Sir J.C. Bose. These are the various methods of measurement of linear growth and stem and radicals. Which methods? Direct methods, horizontal microscope, oxynometer, and chrysographs. It is the methods of stem and radical measured. So, next point is growth rate and types of growth. Growth rate. It is the increased growth per unit time. It is also called as the efficiency index. The growth rate, it is also called as the efficiency index. Rate of growth can be measured by an increase in the size, area of different plant organs like leaf, flower, and fruits. The ratio of changes in the cell number over the time interval, it is dt, or cell number is dn, is called as the absolute growth rate. AGR means absolute growth rate. These are the growth rate, and they also call it as the efficiency index. And the rate of growth can be measured by increase in the size and area of different plants. Means any plant, these organs are like leaf, flower, and fruits are changes in seen. Alternatively, it is the measurement and comparison of total growth per unit time. Moves to the next slide. AGR, absolute growth rate, is equal to dn dt. The AGR when divided, AGR means absolute growth rate when divided by total number of cell present in the medium gives relative growth ratio. It is RGR, means relative growth ratio. Alternative RGR refers to the growth of a particular system per unit time expressed on a common basis, or it is the ratio of growth in the given time or initial growth. RGR is equal to AGR, means relative growth ratio is equal to absolute growth ratio. AGR and RGR are useful in describing the dynamic of cell growth in culture. So next point is types of growth. There are two types of growth: arithmetic growth and geometric growth. First is arithmetic growth. Here, rate of the growth is a constant, and the increase in the growth occurs in arithmetic progressions, that is the 2, 4, 6, 8 centimeters, etc. In this type of growth, the rate of growth is a constant. After mitosis, one of the daughter cell continues to divide and the other cells takes parts in the differentiation and maturations. You also know the mitosis and meiosis. It is equational or reductional division. The arithmetic growth is after mitosis, one of the daughter cell continues to divide and the other cell take parts in the differentiation and maturation. Moves to the next slide. Example is elongation of root at a constant rate. Best explain arithmetic growth. Linear curve is obtained when growth rate is plotted against the time. Arithmetic growth expressed mathematically by an equation. It is expressed as LT is equal to LO plus RT, where LT means length at time, LO means length at a time 0, R means growth rate, T is equal to time of growth. When a graph of length is plotted against the time T, a linear curve and obtained as indicated in the diagram. These are the mathematically by equation of expressed as LT is equal to LO plus RT. LT means length at time T. LO means length at time 0. R is equal to growth rate and T is equal to time of growth. These are the mathematically by equation expressed. Moves to the next slide. Second type is geometric growth. Cell divides mitotically into two. Here both the daughter cells continue to divide and redivide repeatedly. Next, such growth is called as a geometric growth. Here growth rate is slow initially, but later on there is rapid growth at exponential rate. Next, geometric growth can be expressed mathematically by an equation. The picture shows constant linear growth. The geometric mathematical equation is W1 is equal to WO ERT. W1 final size, WO initial size, R is equal to growth rate, T is equal to time of growth, E is equal to base of natural logarithm. These are the geometric mathematical equations. You understand the mathematical equation W1 is equal to WO ERT means W1 is equal to final size, WO is equal to initial size, R is equal to growth rate, T is equal to time of growth, is equal to base of natural logarithm. This picture shows a constant linear growth, the height of the plant and the time. The graph plotted. Moves to the next slide. The picture shows growth rate. It is a diagrammatic representation of arithmetic, geometric growth and stage during embryo development, showing geometric and arithmetic phases. First picture diagrammatic representation shows a arithmetic, second is geometric growth, and third one is a stages during embryo development, showing geometric and arithmetic phases. The picture shows zygotic divided, geometric phases, all cell divided, and this dark blue color is a cell capable of divisions. The dark structure, dark blue color, these are the cell capable of divisions, and the faint color is a cell that lost capacity to divide. These structures are lost capacity to divide, and these are the capacity or capable of cells divides. The formation of zygote dividation, these are the arithmetic phase. The picture shows or diagrammatic representation shows arithmetic, geometric stages or during the embryo development, showing geometric and arithmetic phases. Moves to the next slide. So, we can also observe quantitative comparison between the growth and living system in two ways. Or measurement and comparison of total growth per unit time is called absolute growth rate, means AGR, where is the growth of the giving system per unit time expressed on a common basis per unit initial parameter is called as the relative growth rate, means RGR. Moves to the next slide. The picture shows growth rate comparison. A or B. These are the growth rate comparison. Moves to the next slide. In this above example, two leaves A and B are of differentiate size but show same absolute increase in area in a given time. Both leaves grow and increase their area by 5 cm to produce A and B leaves. A leaf of 5 cm to in size grow 5 cm square per day, then its RGR will be 100%. If the leaf is 50 cm per square in size, the growth rate per day is 5 cm square, then its RGR will become 10%. RGRs mean RGR, it is relative growth rate, and AGR means absolute growth rate. So, we study in this first lecture, the point is growth rate, arithmetic growth, geometric growth, and we study the measurements of the linear growth or stem, radical, direct method, horizontal microscope, oxynometer, and chrysograph. And we study the definition of growth. All points, 4 points study in this lecture. Now, next point study in next lecture. So, thank you.